Okay, so welcome back to this next video in which we are discussing pain and analgesic drugs. Okay, so we're currently in the process of discussing neuropathic pain, which is pain that is caused by damage to the neurons involved in this ascending pain pathway which delivers nociceptive information from the peripheral tissue to the brain. Okay, right. In the previous video, what we discussed is examples of different things which can damage neurons in the peripheral nervous system and neurons in the central nervous system, and which can therefore lead to the development of neuropathic pain if it affects these neurons of the ascending pain pathway. Okay? What we now want to turn our attention to is how these neurons that have been damaged are actually going to start firing action potential spontaneously, because that's what's going to cause the neuropathic pain. These neurons are going to get damaged, and then after they've been damaged, they start firing action potential spontaneously. Okay, now, the mechanisms underlying this spontaneous generation of action potentials are not completely understood yet. Okay, and there's loads of different emerging theories for how it's produced. I'm going to tell you some interesting examples of how it can occur. So I'm going to firstly start off by telling you the major thing that seems to be the overarching theme here, okay, which is the upregulation of NaV1.3 alpha subunits of voltage-gated sodium channels, and the then clustering of these alpha subunits together in the membranes of these damaged neurons. Okay, but I'm also then going to tell you about sympathetically mediated pain that occurs in neuropathic pain, and we're going to do that by looking at neuromas which can form in the peripheral nervous system, and they also play an important role in neuropathic pain, which is caused by damage to the peripheral nociceptive afferents. Okay, so let's start off then with the overarching theme for all neuropathic pain, which is basically that these damaged neurons, whether they're in the peripheral nervous system or the central nervous system, upregulate the expression of a voltage-gated sodium channel alpha subunit called NaV1.3. Okay, so let me remind you then of the structure of a voltage-gated sodium channel. Okay, so here is the membrane. Okay, and then sitting in the membrane, you're going to have your voltage-gated sodium channel. Okay, so the voltage-gated sodium channel, remember, consists of two subunits, an alpha subunit, which is the main pore-forming subunit of the voltage-gated sodium channel, and then a beta subunit, which is the auxiliary subunit, which sits aside the alpha subunit, and together they make the full voltage-gated sodium channel. However, the important thing to note here is that actually you don't need the beta subunit. The alpha subunit on its own here is a perfectly good voltage-gated sodium channel. Okay, the beta subunit truly is an auxiliary subunit that just modifies the properties of the alpha subunit. Okay, now another piece of information is that there are 9 different alpha subunits in humans called NaV1.1 to NaV1.9. Okay, and we discussed earlier how the main forms of NAV expressed in the primary afferent nociceptors, nociceptive neurons, are the NaV1.7, the NaV1.8, and the NaV1.9. Okay, not NaV1.3. Okay, however, damaged neurons start expressing large amounts of this NaV1.3. Now, NaV1.3 is a weird alpha subunit of voltage-gated sodium channels. It's not widely expressed in the adult human that much at all, okay? But it is hugely important in development, okay? So really, we can sort of categorize NaV1.3 as a fetal alpha subunit of voltage-gated sodium channels. It's one that's extremely highly expressed in fetal neurons, okay? But then in the adult, it doesn't seem to be that important. Okay, so it seems that these damaged neurons start turning on a fetal gene. They start expressing an alpha subunit that was hugely used in fetal life, but which isn't usually used in adult human life. Okay, right. And the problem is that these NaV1.3s, they tend to cluster together when they're put into the membrane, so you get huge clusters of NaV1.3 which are really, really dense, and it seems to be these clusters that are then capable of generating spontaneous action potentials. So let me draw a primary nociceptive afferent here. So here is its cell body, here is its peripheral process here, and here is its central process. Let's say it's been damaged, let's say it's been cut here. Okay, it might now start expressing much more NaV1.3, and these might now start clustering in the cell membranes. You might end up with a cluster here, 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 a cluster here. And basically these clusters where you have really, really dense expression of NaV1.3, these seem to be capable of generating spontaneous action potentials. Okay? So why? Well, we can sort of imagine why this might be, okay? Because if you have really dense alpha subunits of voltage-gated sodium channels, then it's likely that any little local perturbation in the electrical potential difference across the cell membrane is going to cause an action potential. So let me explain what I mean by this. If this is the membrane here, and let's now say we have really dense voltage-gated sodium channels in this area here, okay, like so. If you have really dense voltage-gated sodium channels, so each one of these little blobs is representing a voltage-gated sodium channel, then if I make a tiny little flicker in the electrical potential difference across the cell membrane, okay, that might just take the electrical potential difference up to negative 40 millivolts at some little portion, then that's now likely to be near a voltage-gated sodium channel, and now that voltage-gated sodium channel will open, allowing sodium causing the positive feedback, which will spread the depolarization basically, and now all of them will start to open and you'll get the action potential basically. Okay, whereas if we had a more normal expression of voltage-gated sodium channels, okay, let's say they were much more spaced out, and I produce this little local depolarization here, that's not going to open any voltage-gated sodium channels because the effect of the electrical potential difference over here is going to be minimal basically, and the same for here, so these two aren't going to open. So it's when you have these very, very dense collections of voltage-gated sodium channels, you can imagine that any little local perturbation in the electrical potential difference across the cell membrane is then going to cause the generation of an action potential. So you can imagine that things that wouldn't have caused an action potential previously are now going to cause action potentials in these dense clusters of voltage-gated sodium channels that are forming. Okay, so this is one of the key things that is thought to underlie the spontaneous generation of action potentials in these damaged neurons. Okay, and this occurs not just in damaged peripheral nervous system neurons, but also in damaged central nervous system neurons. Okay, so if you get damage to the second order neurons or the tertiary neurons, that could also underlie the spontaneous firing there. Okay, right, so that's the overarching mechanism for neuropathic pain. Damage to the neurons causes them to spontaneously fire, hence you get pain that is caused by the neurons themselves. Okay, what I now want to talk about just refers to damage in the peripheral nervous system, and really it refers to mechanical injury, okay, or potentially amputation of the of an area which then cuts through a nerve when you do the amputation. Okay, and this is the formation of neuromas. So I want to talk about the formation of neuromas and how this can lead to a phenomenon in neuropathic pain known as sympathetically mediated pain. So let's talk about neuromas then. So these are tumours of nerves that are not malignant. Okay, so it's nothing to do with cancer. Okay, it's to do with cutting nerves and then the neurons trying to regrow and forming a tumour as they aberrantly regrow. Okay, so let's do this then. So firstly then, let's say we have some nerve here. So this is not a neuron anymore, this is a nerve. This is a whole cluster of axons. Okay, this is loads of neurons all running in parallel, running back to the central nervous system. Okay, so within this nerve here, you will have the peripheral processes of a huge number of nociceptors. And the idea now is we're going to cut through this, okay, either by mechanical injury, okay, maybe a knife wound of some sort, okay, or amputation, traumatic amputation of a limb. Okay, the nerve is going to get cut through. Okay, now what can then happen is a process known as Valerian degeneration. Sorry, Walerian degeneration. It's not quite as exciting as Valerian degeneration. Okay, which involves these neurons of the peripheral nervous system. So Walerian degeneration, which is also called anterograde degeneration, is a phenomenon that you find specifically in the peripheral nervous system, not in the central nervous system. The central nervous system can sort of do it, but it's really, really slow and it's very half-hearted. And this is one of the main reasons that neuromas really only form in the peripheral nervous system rather than the central nervous system, at least if we're referring to a non-cancerous neuroma here. Okay, right. So what happens in Walerian, I'm going to continue ending up calling that Valerian, Walerian degeneration, okay, is that if we draw an individual axon now, so here is our axon, okay, and its cell body would be over here somewhere, right in the dorsal root ganglion. Okay, potentially. We're imagining it's going to be a nociceptive neuron now rather than one of the other neurons in here. But it could be, you know, any old sensory neuron, or it could be an alpha motor neuron even. Okay, they've all got their cell bodies up at the spinal cord. Okay, so what's going to happen is we're going to cut through it here. Okay, and in the process of Walerian degeneration, what happens is the portion of the axon that is peripheral to the cut, okay, this is all going to degenerate, okay, whilst this bit will remain intact, the bit that's connected to the cell body, which is over at the spinal cord. That's why it's called anterograde degeneration, because you are getting rid of the bit that's in front of, if you like, the bit here, okay, the bit that's further forward. Okay, right, and then what can start happening is this stump here that is more central than this portion, i.e. it's the portion of the axon that was centrally to the cut, okay, it can now start to regrow. So this can now start to regrow and it will extend out. Okay, so what happens in a neuroma is that all of the axons within this nerve are all going to start extending out. And basically, if this goes horribly wrong, what they can form is a great big ball, basically, which contains loads and loads of these growing axon stumps, basically, attached to the original nerve. Okay, and if this was done as an amputation, then these things can become visible as papules on the skin. So let's say if you had your distal phalanx amputated, okay, you could then get little papules on the amputated surface, which are the neuromas becoming visible underneath the skin, basically. Okay, so it's just full of these axon stumps that are trying to regrow, basically. Okay, and this is the site where you can get the beginnings of sympathetically mediated pain. So let me tell you what sympathetically mediated pain is all about. So sympathetically mediated pain refers to pain that is induced by the activation of the sympathetic nervous system. Okay, now how can this actually result? Well, basically there are two mechanisms that are believed to potentially underlie this. Okay, when you get a traumatic injury to the nerve like this, and you get the formation of one of these neuromas, of course there are going to be sympathetic axons running in this, post-ganglionic sympathetic axons running in this nerve, and they're also going to be having axon stumps growing in like so. Okay, and one of the things that is thought to happen is potentially this axon stump of the postganglionic sympathetic neuron could actually join with the stump of a primary nociceptive neuron. Okay, so if we have a primary nociceptive neuron here with its stump extending into the neuroma, the idea is that these two could actually end up connecting to one another, and then whenever you send an action potential down the sympathetic neuron here, it will just continue back up the axon of the primary nociceptive afferent, and therefore you'll get the activation spontaneously of that primary nociceptive afferent in response to the activation of the sympathetic nervous system. Okay, that kind of a formation there where you link one neuron up to another, that is known as an EFAPS. Okay, so this is an EFAPS. Okay, a connection between these two axons. Okay, so that's one thing that can underlie sympathetically mediated pain, the activation of pain when the sympathetic nervous system is activated. The other thing that occurs is that damaged neurons, damaged primary nociceptive afferents, have a tendency, when they are damaged, not just to overexpress NAV 1.3, but also to start aberrantly expressing receptors for noradrenaline, specifically alpha adrenoreceptors, okay, which again are G protein coupled receptors with the characteristic 7 membrane spanning alpha helix structure here. Okay, so these nociceptive afferent neurons here, okay, and I'll just point to this one. This is our primary nociceptive afferent. These can also get alpha-1 adrenoreceptors on their surface. So imagine it doesn't have the EFAPs now. Okay, that's one mechanism for sympathetically mediated pain. This is a separate mechanism now. Basically, what ends up happening is neuromas end up highly sympathetically innervated. Okay, so some of these sympathetic stumps that are growing into the neuroma, okay, they end up remaking axon terminals basically, which will release the neurotransmitter noradrenaline. Okay, and the idea then is that you can form synapses between the stump of the sympathetic neuron and the stump of the primary nociceptive afferent. Okay, so you'd be releasing noradrenaline onto the alpha adrenoreceptors of the primary nociceptive afferent, and that's what would be activating the primary nociceptive afferent. So there are those two different mechanisms underlying sympathetically mediated pain resulting from neuromas. One is the formation of these synapses, the other is the formation of these EFAPSes, and both of them could play a role in the phenomenon of sympathetically mediated pain. Okay, right. So, what we now want to move on to is looking at the drug treatment of neuropathic pain. Okay, so which drugs, which analgesic drugs can be used to treat neuropathic pain? Okay, well, the first example of drugs that I'm going to talk about are the gabapentinoids. Okay, so the gabapentinoids. Now, the main example of a gabapentinoid is the drug gabapentin itself, but there is a related drug known as pregabalin. So the two gabapentinoids are gabapentin and pregabalin, and they're both used to treat neuropathic pain. Okay, so pregabalin is the other one. Okay, so what do these drugs do? Okay, well, these drugs are known to bind to the alpha-2 delta subunits of voltage-gated calcium channels and block them from actually getting incorporated into the voltage-gated calcium channels. Okay, so let me just talk a little bit then about the structure of voltage-gated calcium channels, because we haven't really seen voltage-gated calcium channels before in this video. Okay, so voltage-gated calcium channels, VGCCs for short. Their structure, again, they are huge multi-protein complexes. They have a huge number of different auxiliary subunits. Okay, so I'll start by drawing and labeling up the main pore-forming unit of the voltage-gated calcium channel. Okay, so here is the main pore-forming unit of the voltage-gated calcium channel, and this is known as the alpha-1 subunit this time. And again, just like the voltage-gated sodium channels, where the alpha subunit was one enormous protein, the entire pore-forming unit was one enormous protein. In the case of the voltage-gated calcium channels, again, the alpha-1 subunit, which is the pore-forming unit, is one enormous great protein. So here it is. Okay. Then, in the case of voltage-gated calcium channels, you have a huge number of different auxiliary subunits. So this one that I've already drawn here is what's known as the gamma subunit. So I'll colour that in purple, and that sits alongside the alpha-1 subunit in the membrane. Okay, and then attached onto the cytoplasmic aspect of the alpha-1 subunit of the voltage-gated calcium channel, you have the beta subunit of the voltage-gated calcium channel, which I'll colour in blue here. Okay, and then finally the other subunit also sits alongside the alpha-1 subunit in the membrane, is the alpha-2 delta subunit. Okay, and this is actually made up of two separate structures attached together. Here is the alpha-2 portion, and then the portion which straddles the membrane is the delta portion. And collectively, this subunit is known as the alpha-2 delta subunit. Now, you might be wondering, well, why is that not considered two separate subunits? Why is that considered as one subunit called the alpha-2 delta subunit? Okay, well, the reason is that both of these parts of this structure here actually are made by the same gene. Okay, so I should probably colour them in the same colour. Okay, so basically what happens is you firstly synthesize a pro-protein, basically. You synthesize the alpha-2 delta pro-protein, and then what happens is this is cleaved up. Okay, so you cut it into two portions to make the alpha-2 protein and then the delta protein, and then you reattach them together like so. But this reattachment is different from the way they were originally attached. It's not a peptide link between the two. Instead, it will be links between the R groups of the delta chain and the alpha-2 chain. Okay, and that's why this is considered one subunit, because it is encoded by a single gene. Okay, right. So those are the different subunits of a voltage-gated calcium channel then. The alpha-1 subunit on its own is a functional voltage-gated calcium channel node, but the other subunits moderate its properties, and importantly, in the case of the alpha-2 delta subunit, they control where the alpha-1 subunit, or in fact where the entire voltage-gated calcium channel is actually positioned in the cell membrane, and that's going to become incredibly important. So, these gabapentinoid drugs, gabapentin and pregabalin, they bind to the alpha-2 delta subunit of the voltage-gated calcium channel, okay, and they stop it from being incorporated into voltage-gated calcium channels. So that means that the rest of the voltage-gated calcium channel is now on its own without the alpha-2 delta subunit. Okay, now, why is this important? Well, basically, one of the other changes that has been found in these damaged neurons, okay, is that they increase the number of voltage-gated calcium channels in their synaptic terminals, basically. So if I draw this here, okay, let's say this is the axon terminal of some primary nociceptive afferent which has had its peripheral process damaged, okay? Basically, as well as upregulating NAV 1.3 and being involved in the formation of these neuromas potentially, what is also going to happen is in the axon terminals of these primary nociceptive afferents that have been damaged in this way, you upregulate the expression of voltage-gated calcium channels. Okay, so you put in far more voltage-gated calcium channels than would normally be there. Now, why is that important? Well, what's the function of voltage-gated calcium channels in the axon terminals? Okay, well, they are involved in converting the electrical signal of the action potential into a calcium signal that then drives the exocytosis of synaptic vesicles. Okay, so let me just remind you of how synaptic vesicles are exocytosed. Basically, when an action potential propagates along the axon, it arrives in the axon terminal. The depolarization of the electrical potential difference across the cell membrane that occurs in the action potential causes the voltage-gated calcium channels to open. They allow calcium into the cytoplasm of the cell, and it is this calcium which then causes the exocytosis of synaptic vesicles that are full of neurotransmitter here. if you have upregulated the expression of voltage-gated calcium channels in your axon terminal, that means that whenever an action potential arrives here, you're now going to let in more calcium. More calcium means more exocytosis, more synaptic vesicles are going to be exocytosed, more neurotransmitter is going to be released. That means that you're going to have a bigger effect on the postsynaptic neuron, okay, which in the case of the primary nociceptive afferent would be that second order spinothalamic projection neuron. Okay, so effectively this can sensitize the synapses that the damaged neuron is involved in, basically. Okay, right. So if you give these drugs, gabapentin and pregabalin, and I should just summarize this. This is one of the mechanisms that is believed, therefore, to be important in the pathogenesis of neuropathic pain, that these damaged neurons are now going to induce bigger effects on the next neuron along, okay? And therefore, not only are they going to fire spontaneously, but they're also more likely to induce an effect on the next neuron along, and therefore, that's also going to contribute to increased pain. Okay, so the gabapentinoids then, these work by stopping the alpha-2 delta subunit from incorporating into the rest of the voltage-gated calcium channel. Okay, and what the alpha-2 delta subunit is incredibly involved in is targeting voltage-gated calcium channels to the right portion of the membrane, for instance to the axon terminal membrane. So without the alpha-2 delta subunit, voltage-gated calcium channels don't end up in the axon terminal membrane, basically. Okay, and this reduces the number of voltage-gated calcium channels in the axon terminal membrane. Okay, so because of the fact that these are upregulated in neuropathic pain, these drugs are supposed to be specifically effective against these neurons that have been damaged and which have upregulated the expression of voltage-gated calcium channels in their axon terminals. Okay, it's important to state that you're not going to give these drugs in high enough concentrations such that they completely stop all voltage-gated calcium channels being in the axon terminals in all neurons. Again, that would kill you. Okay, you're going to give it in a dose that's going to lower the number of voltage-gated calcium channels that actually end up in the axon terminal membranes by taking out quite a few alpha-2 delta subunits. Okay, right. So that's gabapentin and pregabalin, the gabapentinoids. Okay, the next drug that I would like to discuss is carbamazepine. Okay, which is the first line of treatment for a form of neuropathic pain known as trigeminal neuralgia. Okay, so carbamazepine. Okay, so firstly, let me tell you that carbamazepine is an anti-epileptic drug. Okay, so this is an anti-epileptic. It's used to stop epileptic seizures. Okay, right, whoops, missed out an I there. Anti-epileptic. Okay, right. Now, how does carbamazepine work to target epilepsy? Well, basically, it's the same mechanism as we have seen for the local anaesthetics. And in fact, we're going to see another class of drugs later on, the anti-dysrhythmics, which are involved in treating dysrhythmias in the heart, which again have this same mechanism as the local anaesthetics. Okay, carbamazepine, just like lidocaine, is a use-dependent blocker of voltage-gated sodium channels, and through that same mechanism that we've discussed for lidocaine, it stops neurons from firing which are trying to fire too rapidly, but not neurons that are not trying to fire too rapidly. Okay, so again, just like local anesthetics, anti-epileptics, or at least most anti-epileptics, the ones which target voltage-gated sodium channels, not just carbamazepine, other anti-epileptics as well, the way they work is by blocking neurons that are firing too rapidly. Now in epilepsy, just to draw a little picture of the brain here, in epilepsy there is a bad patch of neurons, okay, which is known as the epileptogenic focus. Okay, so this is an I'm spelling awfully, epileptogenic focus. Okay, and this area starts discharging action potentials at a ridiculous rate to all other areas of the brain, basically. And this then is what leads to the epileptic attack, basically. You know, if you start sending a huge amount of rapidly firing action potentials to, let's say, the motor cortex, then you're going to start getting uncontrollable movements, basically, and that's what causes the convulsions that are usual in many epileptic attacks. Okay, so in epilepsy then there is this epileptogenic focus which contains neurons that have had bad things happen to them and now are firing at ridiculously high rates, and which leads to the epileptic attack. Okay, so the idea behind how carbamazepine can then be used to treat this is that it's going to block these neurons from firing because they're trying to fire too rapidly, but it's not going to fire all of the good neurons of the brain which are behaving well and aren't firing anywhere near as rapidly. Okay, and that's how it can stop the epileptogenic focus without killing you, basically. Okay, right, so carbamazepine can also be used to treat neuropathic pain, okay, again because it's going to, presumably at least, because it's going to block neurons that are firing very rapidly, which these spontaneously firing damaged neurons are going to be doing effectively. Okay, however, the weird thing is that carbamazepine can only, well, it's only effective against a certain form of neuropathic pain, and this certain form of neuropathic pain is trigeminal neuralgia. Okay, so it's not effective against most cases of neuropathic pain, only trigeminal neuralgia. Okay, so let me explain what happens in trigeminal neuralgia. Okay, so trigeminal neuralgia affects the nociceptive primary afferents in the trigeminal nerve. Okay, so the other name for the trigeminal nerve is cranial nerve 5. CN stands for cranial nerve here. Okay, and basically the pathology of trigeminal neuralgia is believed to involve the trigeminal nerve getting compressed by an artery, basically, at the point that it enters the brain. Okay, so let me just tell you where the trigeminal nerves actually enter the brain. Okay, basically they enter the brain at the level of the pons. Okay, so what I'm now going to do is draw the pons from the front. Okay, so I'm now drawing the brainstem from the front. We have seen the brainstem from the side many times, but we haven't seen it from the front before. So here's the pons viewed from the ventral aspects. We're looking at it from the front now. And then underneath you'll have the medulla, and then underneath that you would then have the spinal cord down here. Okay, right. Now the trigeminal nerves are very large cranial nerves that come off the pons like so. And they carry a huge number of sensory fibers into the brain, basically. Okay, so these are the trigeminal nerves, and of course you have two trigeminal nerves. You have a left trigeminal nerve here and a right trigeminal nerve here. Okay, right. So in trigeminal neuralgia, what is believed to happen is the trigeminal nerves get compressed by an artery that is located very nearby where they enter the pons, basically. Okay, in fact, it's located just above them. So let me talk about this artery. So the first thing to say is that on the ventral surface of the pons, running right down the middle here, is an extremely important artery known as the basilar artery. Okay, so this is the basilar artery. And then near the top of the basilar artery, as it approaches the top of the pons, two important branches come off. One on the right-hand side there, and one on the left-hand side. Okay, and these are the superior cerebellar arteries. Okay, so I'll just label these up in a moment. So this is the basilar artery, and these two branches here, these are the superior cerebellar arteries. So this one is the left superior cerebellar artery, and the other one is the right superior cerebellar artery. Okay, and these will supply, these will supply the top of the cerebellum with blood, basically. Okay, because the cerebellum will be behind all of this, and these will go back and supply the top of the cerebellum. So let's just colour these in. So here we have the basilar artery here, and then we have the superior cerebellar arteries coming off there. Okay, right. And basically, trigeminal neuralgia is believed to occur because the superior cerebellar artery compresses the trigeminal nerves at the points that they enter the pons here, and that then leads to damage to fibers entering here. And if those fibers are nociceptive fibers, then that can then lead to those fibers starting up firing spontaneously, and that then leads to neuropathic pain. Okay? Carbamazepine is very effective in treating trigeminal neuralgia, in stopping these nociceptive afferents, which are firing spontaneously due to this compression, which has damaged them, from firing that rapidly, basically. And its mechanism is by stopping neurons which are firing too rapidly, and it uses that same mechanism that we discussed for the local anaesthetics. Okay, whilst we're on the topic of of drugs which stop neurons from firing too rapidly, other treatments for neuropathic pain which stem beyond just trigeminal neuralgia, these other treatments are capable of treating a much wider range of neuropathic pains. Okay, you can also use lidocaine, the local anaesthetic, okay, which can be delivered systemically to treat neuropathic pain. Okay, so again, that works through the mechanism that we've discussed. It's going to stop neurons which are firing too rapidly, and hence it will stop these damaged neurons which are discharging spontaneously at a very high frequency. Okay, another drug that I want to discuss is actually an example of an anti-dysrhythmic. Okay, and this is the drug flecainide. Okay, so let me explain why anti-dysrhythmics also include drugs with this same mechanism of stopping action potentials from firing too rapidly. Okay, so this is an anti-dysrhythmic drug. Okay, so flecainide has this same mechanism as lidocaine and carbamazepine, the anti-epileptic. Okay, now why is this useful as an anti-dysrhythmic, okay, in treating problems with the electrical activity of the heart? Well, basically, in certain dysrhythmias, okay, and I'll just draw a little picture of the heart so that I have something to point at. So here is the right atrium here. Here is the right ventricle here, then we've got the left ventricle here, the left atrium here, and then the aorta coming off there, and then you should have the pulmonary artery there, but we'll say that's the semilunar valve that I've put in there. Okay, right. So basically, in dysrhythmias, one key example of a dysrhythmia is what's known as a tachycardia. And in a tachycardia, the heartbeat is far too rapid, okay? And what can drive a tachycardia is that some badly behaving cardiomyocytes, either in the sinoatrial node, which is the place where you are meant to have the action potential originating from, or elsewhere in the heart, they are firing action potentials too rapidly. Okay, so some badly behaved cardiomyocytes are now firing action potentials too rapidly. Okay, and this then is spreading to all the other cardiomyocytes, and therefore is causing the heart to contract far too rapidly, and that's what's known as tachycardia. Okay, so if we give a drug that is going to block cardiomyocytes firing action potentials too rapidly, and you can do this in exactly the same way as we have done it for neurons, okay, because the same voltage-gated sodium channels are important in the heart, okay, for the upstroke of the action potential, then we can stop the cardiomyocytes that are firing action potentials too rapidly without stopping all the other cardiomyocytes which are firing action potentials at a normal, reasonable rate. Okay, and therefore wherever these badly behaved cardiomyocytes are, whether they're actually in the sinoatrial node and are causing the sinoatrial node to discharge too rapidly, or whether they're at some ectopic locus, maybe over here. Okay, so this would be called an ectopic focus, if so. Then we can stop those firing too rapidly, and therefore we can stop that portion driving the heart to beat too rapidly, because if that portion is now suppressed, then the normal healthy cells of the sinoatrial node can go back to discharging action potentials at their normal rate, and therefore they won't be in this overdrive, basically. They will now be driving the heart normally, and your heart rate will go back to normal. So I'll stress that again. What happens is you get some badly behaved neurons either in the sinoatrial node or in an ectopic focus that then discharge action potentials far too rapidly. That then spreads throughout the entire heart and causes the heart to beat far too rapidly. Okay, if we can silence these badly behaved neurons, then the neurons in the sinoatrial node that are still working properly, they will now be able to fire their action potentials at the normal rate, and therefore the heart rate will go back to normal. Okay, so we give a drug that will only block those patches that are firing far too rapidly and not the portions that are firing at the normal heart rate. And that's the origin of this drug flecainide and how it can be used to treat the tachycardia induced by these badly behaved cardiomyocytes. Okay, right. So lidocaine and flecainide, they both also decrease the ability of neurons that are firing too rapidly. So they block neurons that are firing too rapidly. So flecainide doesn't just have its effect on cardiomyocytes, it will also do the same as lidocaine. Okay, so it too can block these damaged neurons which are firing action potentials far too rapidly, basically, and therefore it can also be used to relieve neuropathic pain. Okay, so we'll call it there for this video, and in the next video what we'll discuss is how the tricyclic antidepressants and the serotonin and noradrenaline reuptake inhibitors, the SNRIs, can be used to treat neuropathic pain. And then we'll mention the opiates